Hey guys, so I hope you're all doing well. Today I wanted to talk to you about the pentose phosphate pathway. We're going to look at when this pathway occurs, the main components, and kind of what it produces. So I hope you're excited and let's get started. The pentophosphate pathway, or PPP, or phosphogluconate pathway, or hexose monophosphate pathway, you can see it called many ways, is one of the pathways of our metabolism, and it's going to help us produce a certain number of products that can be integrated into other pathways. So as you know, in metabolism, every pathway, well, most pathways are linked with each other. As we're going to see, this pathway is super important for creating nucleotides, coenzymes, DNA, and RNA, but it can also create glucose 6-phosphate, which can help to bring us energy. This glucose 6-phosphate can then be integrated into glycolysis to create the energy, as you very well know. But we are going to get into the details of that in a little bit. This pathway occurs in the cytosol and is going to be called on every time we need the products that are created by it that we're going to go through later on. First, let's look at the composition of this pathway. As you can see, I've marked down that there are two phases for you. On the left, the non-oxidative phase, and on the right, the oxidative phase. The main components of the pathway are the ones that are marked in black, but as you can see on the side, whether it be in orange or purple, there's a lot of side reactions going on that are absolutely essential to make sure that the reactions in black go smoothly. So this includes enzymes or oxidation, things like that. But we're going to go through it one step at a time. Okay, so let's start with the oxidative phase. We start out here with glucose 6-phosphate, which you probably know can be derived from glucose using hexokinase to add a phosphate. So a kinase is an enzyme that adds a phosphate, and hexokinase means it adds a phosphate on the 6th carbon, so glucose 6-phosphate. And glucose, we just got it from anything we may have eaten, right? So we have our glucose 6-phosphate. Now, what's going to happen to it? Well, glucose 6-phosphate is going to be transformed, it's going to be oxidized into 6-phosphogluconate. And how do we know it's going to be oxidized? Well, because as you can see on the right of it, the orange reaction, NADP plus is going to be reduced to NADPH. So when you have a reaction where one element is reacting with another and the first is being oxidized, well then the other one is going to be reduced. So NADP plus is reduced, therefore glucose 6-phosphate is oxidized. At the same time as this, GSSG is going to be transformed into 2-GSH, which is called glutathione reductase. We're going to talk about that component a little bit later. Next, 6-phosphogluconate is going to undergo pretty much the same process. It's going to be oxidated, but it's going to release a CO2 molecule. So very important, the pentose phosphate pathway, the oxidative phase releases a molecule of CO2. Once again, NADP plus is going to be reduced into NADPH, but here we're going to have the transformation that's coupled with it is going to be precursors that are going to be transformed into fatty acids or sterol, which is basically a steroid alcohol. This process is called reductive biosynthesis. It's also something we're going to see later, but as you can see, it's very useful. We are transforming these precursors into fatty acids, which we know are very useful for us. All over our body. Loads of cells use this. So we transform the 6-phosphogluconate through oxidation and a loss of the CO2 molecule into ribulose 6-phosphate. Now, what's going to happen here is we're going to transform it into ribose 5-phosphate. This is kind of the intermediate step between the oxidative phase and the non-oxidative phase. Why is ribose 5-phosphate so important? Because it helps generate nucleotides, coenzymes, DNA, and RNA. So it's a really useful building block for downstream processes. Okay, so we arrived at our middle point, the ribose 5-phosphate, and now we're going to go into the non-oxidative phase, so the part that is in purple, and we're gonna make a whole loop right up to the glucose 6-phosphate, which is where we started. So what happens to ribose 5-phosphate? Ribose 5-phosphate is going to be transformed by transcatolase, which is an enzyme, into acetoheptuose 7-phosphate. All right? But at the same time, ribose 5-phosphate can be transformed by the successive enzymes of isomerase and epimerase into xylulose 5-phosphate. So the P each time is phosphate, right? And the 5, 6, 7, the number, that is the position that this phosphate is on our molecule, which has a carbon chain. And by the same enzyme, transcatolase is going to transform cellulose 5-phosphate into glyceraldehyde 3-phosphate. All right? And you should recognize the name glyceraldehyde 3-phosphate as it is one of the enzymes. It is one of the molecules in glycolysis. So glyceraldehyde 3-phosphate is the product of the fourth step of glycolysis. So that's great, right? We can now use our glyceraldehyde 3-phosphate if we want to directly go into glycolysis to produce energy. But let's not look at that right now. From acetoheptuose 7-phosphate, it's going to get transformed into fructose 6-phosphate. And this is by the enzyme called transaldolase. At the same time, the glyceraldehyde 3-phosphate that we were talking about before is going to be transformed by the same enzyme into erythrose 4-phosphate. Now, here's where it gets kind of messy. This erythrose 4-phosphate can be transformed into fructose 6-phosphate. This is kind of a separate pathway that we can take to get to the final thing, which is glucose 6-phosphate. All the way on the left, you can see the cellulose 5-phosphate, maybe residues from what we had before, is going to be transformed into glyceraldehyde 3-phosphate, just as we saw with the use of transcatolase, but this time using 3 successive enzymes, which is triose phosphate isomerase, aldolase, and fructose 1,6-biphosphatase. It's going to be transformed into fructose 6-phosphate. From there, using phosphohexose isomerase, we can go all the way back to glucose 6-phosphate. So it does look a bit messy, but all it means is that there are multiple pathways in which we can get back to glucose 6-phosphate in this whole pentose phosphate pathway kind of thing. And what's really useful is we see that a lot of the same molecules are being produced, and so the more a molecule is being produced in a pathway, the more the pathway wants to produce this molecule. Alright, so that was kind of a really big overview of the actual enzymes. You shouldn't worry too much about the actual enzyme names as long as you kind of understand what is going on. And what I really want to try to communicate to you here is what is the point of this pathway? What are they trying to produce? And so that's what we're going to look at right now. So I've written little notes for you on the bottom. On the right side, I've written you the chemical equation that describes the oxidative phase of the pentophosphate pathway. On the other side, I've kind of written additional information. These are reversible reactions, first of all. The function of the non-oxidative phase is to recycle pentose phosphate and hexose phosphate. As you can see, the oxidative phase produces NADPH, so the oxidative phase and this pathway will be used a lot in cells that need a constant regeneration of NADPH. Now, NADPH is not to be confused with NADH, which is a molecule that's used more in glycolysis and things like that. At the very, very top, I've written down the rates of NAD plus to NADH per cell, which is about 1,000, and the rate of NADP plus to NADPH is only 0.1. So you can see that NADH wants to accept electrons, so you'll see it in, NAD plus wants to accept electrons, so you'll see it more in the electron transfer chain, while NADP plus, NADPH wants to donate its electrons, which is important, means it's important in the anabolic pathways, and so in its oxidative phase. So that's kind of like a little trick, so you know which one is used where. Now, the pentose phosphate pathways function differently depending on the needs of the cell, so we're going to go through 3. For proliferative cells, so this can be those of the intestine, of the skin, or immune cells, they're going to need to, well, proliferate, so synthesize nucleotides. So that's what they're going to use it for. Remember, ribose 5-phosphate is used to synthesize nucleotides. For secretory cells, this could be in the adrenal glands, they need hormones, or in the liver. They are going to need to produce NADPH to help them accomplish that. So they are also going to go through the oxidative phase, right? And cells that have a high respiratory potential, muscle cells or fat cells, which we call brown adipose cells, they're going to need energy generation. So they're going to use the glucose 6-phosphate or the glyceraldehyde 3-phosphate produced by this pathway so they can integrate glycolysis and produce energy. Please note that there is no ATP that is produced or consumed in this pathway. It is ATP free. Okay, so I wanted to do a little segment on NADPH. So NADPH stands for nicotinamide adenine dinucleotide phosphate. And I've drawn the molecule for you here and labeled the corresponding parts. What are this molecule's functions? So there are two main ones. The first one is reductive biosynthesis. Remember, we talked about this early on. So NADPH is a molecule that's very rich in electrons, right? It can donate it to become an ADP+, so it's very rich in energy. For example, it's going to be used to synthesize fatty acids. So that means there's going to be a high concentration in the liver, adrenal glands, or reproductive organs. So reductive biosynthesis means that we're using reductions, so we're going to reduce NADPH to form NADP+, and doing that, we're going to synthesize biological molecules. All right, and the second really important function that NADPH has is that it protects against oxidative stress. So this is what I talked to you about before. The reduced glutathione, or GSH, protects the cell by neutralizing hydrogen peroxide, so transforming it into water, as well as free radicals such as OH-. So these free radicals and hydrogen peroxide are really toxic to the cell. And so reduced glutathione is going to help doing that, right? And the regeneration of reduced glutathione, or GSH, from its oxidized form, GSSG, so this is exactly what we saw before, needs NADPH. And NADPH is actually regenerated by glucose 6-phosphate dehydrogenase. You can clearly see this is very important because if we have less than 2% of residual GCPD in our cells, we can have severe clinical symptoms. So this is very important. Okay, so I hope that this was clear. Please make sure to let me know if it wasn't, if you have any questions, if you would like me to cover any other topic, whether that be in physics, math, chemistry, or biology. I would love to answer you. Until then, as always, stay safe and happy learning.